All right, are there any questions? Okay, so last time we were looking at the structure of proteins, all right, protein structure and function, and I mentioned towards the end of the class that there are, although the structure of a given protein might be very complicated, but there are a few different kinds of motifs, all right, that occur over and over again, right, and that actually help us in studying the structure of a protein. So two of them are the alpha helix and the beta sheet, right, and both of these structures, they result from hydrogen bonding between NH, right, and C double bonded with O. Remember, proteins are made up of units that are called amino acids, right? Amino acids have an amino group on one side, right, that is NH2, and a carboxyl group C double bonded with OH on the other, right? And then you link the amino acids together by what kind of bond? Peptide bonds, okay. Now when you have a peptide bond, you will have an NH sticking out and you will have a C double bonded with O sticking out, okay. And that is independent of the particular side chain that you have, okay. Now remember, again, hydrogen has got a low affinity for electrons, right. Oxygen has got a high affinity for electrons. So the hydrogen in the NH, right, could hydrogen bond with the oxygen in the C double bonded with O. Okay, and that's independent of the side chains that you have, right? And that's what gives rise to these two structures called alpha helix and beta sheet, right? So therefore, these two structures are quite common because it doesn't depend on the actual side chains that you have. It just depends on the polypeptide backbone, right? And so these structures can be formed by many different amino acid sequences. So now let's take a little bit more detailed look at the alpha helix and the beta sheet, right? So this is the diagram of a typical alpha helix, all right? So every fourth peptide bond, okay, there will be a hydrogen bond that will be formed between the hydrogen in the NH, right, and the oxygen in the C double bonded with O, right? And so you will get a helical structure like this, and the atoms are colored differently. Like hydrogen is colored in white, okay, carbon in black, all right, oxygen in red, right? And so you can see how the bonding happens. So how is an alpha helix generated? It is generated when a single polypeptide chain turns around itself to make a rigid cylinder. A hydrogen bond is formed between every fourth peptide bond, right? And this gives rise to a regular helix with a complete turn every 3.6 amino acids. So you have to do the detailed math. I haven't done it, but you can do the detailed math to arrive at the number of 3.6. Now sometimes you will have alpha helices that wrap around each other, right, to form a particularly stable structure which is called a coil, right. So the alpha helix itself is like a coil, right. You could have two alpha helices that fit into each other, right, and they form what is called a coil. So that's another structure that you see quite often, right. Now the figure on the next slide is going to show a transmembrane protein. That means a protein that crosses a membrane, right. Remember, the cell membrane is hydrophobic, right? But on either side you have basically an aqueous environment, right? So you can have proteins where, so a transmembrane protein will have to cross that hydrophobic barrier, okay? And usually it does that in the form of an alpha helix, right? So if you look at the next picture, right? So here you have a lipid bilayer. Again, I mean, by now you guys know, right, why it's a bilayer, right? The portion that hates water stays away from it. The portion that likes water will stay in contact with it, right? So this is a typical lipid bilayer, and you could have an alpha helix that is crossing that lipid bilayer, right? And in order for that crossing to be feasible, see that alpha helix has got different side chains. Some of them are hydrophobic, some of them are hydrophilic. The hydrophobic ones, they will face the lipid bilayer because the interior of the lipid bilayer is hydrophobic, right? The hydrophilic ones will be in the inside of the helix, right, away from the lipid bilayer. So again, no major surprises. Any questions? Now the beta sheet, again, it is formed by hydrogen bonding, right, between the hydrogen on the NH, right, and the oxygen on the C double bonded with O, right. So if you look at the structure, and again, I'll first show the picture because that is going to convey a lot more information. So you could have a situation like if you look at the picture on the right, right, this is what would be called a parallel beta sheet, right. So you have a polypeptide chain, part of it is laid out like this from left to right, right. The next one again from left to right, right. In between there is a connection, right. And then there will be hydrogen bonds that will be formed between the NH, right, and the C double bonded with O, right. So this would be a parallel beta sheet on the right, right? And on the left, it would be running anti-parallel because you can see the arrow is pointing from left to right, right? And then the next one is going from right to left. Okay. So let's just go back to the text over here. So beta sheets can form either from neighboring polypeptide chains that run in the same orientation. I showed that to you in the diagram. So these are parallel chains, or a polypeptide chain that folds back and forth upon itself, that is forming anti-parallel chains. Right? And these sheets are held together by hydrogen bonds that connect the peptide bonds in the neighboring chains. Right? Again, what kind of hydrogen bond? NH, C double bonded with O. The H and O, right, will form a hydrogen bond. Right. Now both these types produce a very rigid structure, and I've already shown you the figure, so we can skip this slide and go to the next one. Now what are the levels of organization in proteins, right? So the structure of a protein can be studied at 4 different levels, right? The first one is the primary structure, which corresponds to the amino acid sequence, right? A protein is made up of amino acids that are linked together by peptide bonds. So the first level of studying proteins would be to look at what are the amino acids, right, that make up that protein, right? Then you look at the secondary structure, which corresponds to the existence of alpha helices and beta sheets. Okay, I showed you already what an alpha helix is, what a beta sheet is, right? So what are the alpha helices and beta sheets that are there in this protein, that make up this protein? That would be the secondary structure. Then the tertiary structure will correspond to the 3-dimensional conformation, because ultimately at the end of the day you have the alpha helix, you have the beta sheet, then you have all these side chains interacting with each other. But at the end of the day, it's the 3-dimensional conformation of the protein that is the most important, right? And that is basically the study of that 3-dimensional conformation corresponds to the tertiary structure. And the quaternary structure will correspond to studying the complete structure for proteins that are made up of more than one polypeptide chain. There are proteins which are made up of more than one polypeptide chain. For example, hemoglobin is made up of 4 different polypeptide chains. Okay, so if you're going to study the structure of hemoglobin, that would involve looking at the quaternary structure. Any questions? Now sometimes in a polypeptide chain, parts of it might independently fold up into different shapes, right? And you'll be calling that a protein domain. So a protein domain is produced by any part of a polypeptide chain that can fold independently into a compact stable structure, right? And a domain usually contains between 50 and 350 amino acids, and it is the modular unit from which many larger proteins are constructed. Now, see, there are only 20 amino acids that occur in nature, right? So if you are looking at a protein, in every position, there are 20 possibilities, right? So if you had a protein which was, let's say, 250 amino acids long, then you have 20 to the power of 250 different polypeptide chains, right? I mean, we know that from permutation combination, right? However, only a small fraction of these would adopt a stable 3-dimensional conformation, right? Now proteins that don't have a single stable conformation, they are not biologically useful, because as I said, in biological function, shape is everything. If you don't have the right shape, you're out of business, all right? So proteins which are not useful, okay, I mean, in theory you can form lots of different proteins. If they don't form a stable 3-dimensional, if they don't have a stable 3-dimensional shape, right, then they are useless. So nature has eliminated them, right? We don't see them around. So by the process of evolution, those proteins are gone. The ones that don't have very precise shapes, right? Precise 3-dimensional conformations. In fact, a present-day protein will have a single stable conformation with properties that enable it to perform its function. As I have emphasized in an earlier lecture, hemoglobin has got exactly the right shape for binding oxygen. Okay, that's its function. Okay, it has to have that shape. Okay, so I mean, this is some kind of natural selection that determined that hemoglobin is the molecule that's going to do the job. So hemoglobin has the right 3D shape that enables it to bind oxygen atoms and carry them from the lungs to the tissues. If you go and disrupt the shape a little bit, actually hemoglobin is made up of 146, actually the beta, it has got 2 subunits. Okay, one of the units has got 146 amino acids. You go and change one of those amino acids, you've lost function, right? And if somebody has got defective hemoglobin, that person could have the disease that is known as sickle cell anemia, right? It can be life-threatening too. Next, let's talk about protein families. So once a protein evolves into a stable conformation, its structure could be modified slightly to enable it to perform new functions, right? Like we have different copies of hemoglobin in us, okay? I mean, we have copies of hemoglobin that are used at the adult stage, right? At the, uh, infant stage and so on. Okay. In fact, there are 5 different copies of hemoglobin. So they are related, they're in the same family, but they're altered a little bit, right? So that they are especially well suited for the function at the particular stage of life they are meant for. Okay. So these kind of proteins would be grouped into what are called protein families, and members of each such family display many similarities, including their three-dimensional conformation. Right? The three-dimensional conformation will be very similar, but in addition there might be some slight modification that will adapt that protein for, maybe that particular stage in life, right, or a particular environment, and so on. Now, large protein molecules often contain more than one polypeptide chain. I've said that, hemoglobin has got more than one polypeptide chain, and each polypeptide chain is called a protein subunit. Again, I mean, as I'm going along, there are things in blue, okay? That's terminology. You should be noting that as we go along, right? Not right now, right? But when you're studying for the exam, or at least to get something out of this course. Now, the subunits will bind to each other by weak, non-covalent bonds. When the polypeptide chains are identical, we have what are called dimers, right? 2 polypeptide chains that are forming a protein. If there are 4 identical polypeptide chains, then you will have what are called tetramers, right? You can even have octamers and so on. But there are proteins which can also contain 2 different types of polypeptide chains, and an example of that is hemoglobin, right? Hemoglobin contains 2 identical what are called alpha globin subunits and beta globin subunits, right? And we'll talk more about hemoglobin later on in the context of sickle cell anemia and so on. Okay? Now, proteins can assemble into filaments, sheets, or spheres. As I said, proteins are extremely versatile molecules, right? They are responsible for most cellular functions, so they can form all kinds of shapes that are particularly well suited to that function, right? Now, protein units can also bind to each other to form a very long helix or a circular ring, as we will see on the next slide, right? So you could have a free protein subunit that looks like shown over there on the left, right? Then you can see that, 2 units like that could bind together, right? It's like fitting into each other to form what is called a dimer, right, at the top. In addition, they could be binding like, you could have a head and a tail, and they could be, binding to each other, form a structure like a helix, right? That's the picture in the middle of the page, right? And at the bottom, you can see that units could be binding to each other to produce a ring, right? So lots of different possibilities, and they're well suited for a particular biological function. Now, structures such as viruses and ribosomes. Okay, ribosomes we have not encountered as yet, but ribosomes are protein-making machinery. We will talk about them when we have covered DNA, RNA, and then we do transcription, translation. We'll talk about ribosomes. But these structures, such as viruses and ribosomes, they are built up from a mixture of one or more proteins, right, and RNA or DNA molecules, right. Like as I said before, a virus is not a living object, right? It's just some protein, some RNA or DNA that is encapsulated inside a protein code. All right, that's all there is to it, right? But it is going to hijack the replication machinery of the cell and make many, many copies of itself. All right, and in the process kill the cell. Okay, that's how they make you sick. Right? Now, all these structures like viruses and ribosomes, they can be isolated in pure form, and they can be dissociated into their constituent molecules, right? Whether they are proteins, whether they are RNA or DNA. But if you again go ahead and mix them together, you will see that these structures can spontaneously assemble. Right. A helix is a common motif that is found in many biological structures, and it results from similar units connected end to end. Right. We have already seen the alpha helix for proteins, but I'm saying even proteins, other proteins also, units could link together, as I showed you in the earlier picture, right, to produce a helix. Now, some proteins have got elongated fibrous shapes, such as alpha keratin, which is a protein that is found in hair. Then there are other, and again, for us right now, this is all like Greek, okay? There is another example is collagen. Another example is elastin, right? So collagen is found in the skin, right? But we are not worried about the function of all these different proteins, right? Now, proteins outside the cell, that is extracellular proteins, are often stabilized by covalent cross-linkages, such as the disulfide bonds between cysteine side chains. Right? Anybody remember what was the structure of cysteine? I'm not asking you to be responsible to remember that, but I covered that. Okay, what was specific in the case of cysteine? Can you remember what did you see in the side chain of cysteine? What? Yeah, yeah, yeah. So it has sulfur. Yeah, 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 yeah. So let's look at the picture on the next side, on the next slide, all right? So if you're looking at a cysteine side chain, okay, that's what you have in the picture on the left, right? There are two cysteine side chains, all right? So you have sulfur and you have hydrogen, right? Now in the aqueous environment of the cell, there is an excess of hydrogen, right? So this is the structure will be what you see on the left. But when the particular protein is being exported from the cell, right, hydrogen is becoming a limiting factor, and you could have a disulfide bond formed between the two sulfurs, right? So this is like an atomic staple, right? It's not going to determine the three-dimensional conformation, but it's like you've made the conformation and then you stapled it, right? So when it's exported from the cell, you would have the disulfide bond that you see on the right. Right? So that's what this stuff is talking about. Extracellular proteins are often stabilized by covalent cross-linkages such as the disulfide bonds between cysteine side chains, and they serve as atomic staples to stabilize the three-dimensional conformation of the protein outside the cell. And inside the cell, such staples are not needed and hydrogen ions are readily available. So what they will do is if you inject it inside the cell, hydrogen will get attached, the disulfide bonds will be gone. Okay. All right, any questions? Next, I will talk about protein ligand interactions. Again, this is terminology, right? Maybe you're seeing it for the first time, but biologists talk about it as though it's like ABC, or these terms like ligand, substrate, and so on. Okay, that I will cover. So proteins bind to other molecules, which are called their ligands. To ligate means to tie. Okay, so call their ligands with great specificity, right? Each protein, it will pick up its partner with great specificity. Okay, it's not going to bind to anything, right? So the ligand must fit precisely into a groove on the protein surface, the three-dimensional conformation. This allows for the protein and its ligand to be linked together strongly by large numbers of weak covalent bonds, right? And facilitating their interaction. So this perfect kind of fitting enables a protein to pick out its ligand or partner in a crowded environment. And the region of a protein that associates with the ligand, because you're looking at the three-dimensional conformation, okay, the portion that associates with the ligand, that is called a binding site. Again, terminology that we have to pick up, as we go through this material. So ligand and binding site. Now we all know about antibodies, okay, and we know about immunotherapy, right? We get shots all the time, right? So how does that work? Well, antibodies or immunoglobulins, right? These are a class of proteins that have a highly developed binding capacity. So they are produced by the immune system in animals in response to foreign molecules, right? So each antibody is going to bind to a particular target molecule very tightly, and either it will inactivate the target directly or it will mark it out for destruction. It will label it as a bad guy. Okay, that's to be taken out, right? So what do antibodies look like? Antibodies are actually Y-shaped molecules, right? They look like this, a typical antibody molecule. So you have a number of polypeptide chains. So you have the light chain, okay, that is at the bottom of the, like it's the lower one, okay. It's marked as the light chain over there. So you have two light chains, right? And then you have a heavy chain. Heavy chain means it's longer, right? But notice that at the top, right, you have these antigen binding sites, okay? For each antibody, it has exactly the right shape to accommodate the appropriate antigen molecule, right? So if it's an antibody against some virus or something, it'll have exactly the right shape so that the virus will fit in over there exactly, right? Like a hand in a glove, right? So antibodies are Y-shaped molecules with two identical binding sites. Each site is complementary to a small portion of the surface of the target, which is called the antigen molecule, right? So an individual animal can make billions of different antibody molecules against billions of targets, right? So each antibody molecule has got a distinct antigen binding site. Antibodies defend us against infection in at least two different ways. First, antibody and antigen aggregates. So when the antibody binds to the antigen, right, these are ingested by the phagocytic cells. The phagocytic cells are like the trash cleaners in your body, okay? They are going around and getting rid of trash, right? So if something is marked with an antibody, that will be taken up by the phagocytic cells, number one. The second thing is that antibody-coated bacteria or viruses, these are killed by special proteins in the blood because these have been marked as the bad guys, right? So they are taken out, right? Now, how does immunotherapy work? Well, antibodies are made by a class of white blood cells, which are called the B cell lymphocytes, or they are also called the B cells, right? So each B cell carries a different membrane-bound antibody molecule on its surface, right? So that Y is bound on the membrane of the cell, of that B cell, right? Now, so each B cell carries a different membrane-bound antibody molecule on its surface that serves as a receptor for recognizing a specific antigen, right? So each antibody is specific to a particular antigen, right? Same antibody is not going to work for different antigens, right? So when the appropriate antigen comes and binds to the receptor, right? What is going to happen is that the B cell is stimulated to divide. And I don't know, those of you that attended my seminar last week, all right, I talked about the cell dividing, how something comes and binds. So the same thing happens, right? So when the antigen comes and binds, right? So it's the stimulus that the B cell needs to divide, right? So the B cell is going to divide, and and it is going to produce large amounts of that same antibody. Okay. So this phenomenon can be used to raise the level of antibodies in animals because you give a small amount of the antigen, right? It binds to the B cell. The B cell is going to divide, right? Produce large amount of that antibody, right? So now you have, the power or the strength to basically fight that same antigen if it were to invade your body in the future. Yeah. Yeah, you have a question.
How our body knows that which kind of virus will attack us and have that antibody inside ourselves? Because it said that
No,
some
no, no.
viruses attack our
No,
body.
our body does not know that. Okay, but
How beforehand has that kind of we have this kind of antibody in our
uh,
body?
probably it's some of it is inherited. Okay, I mean like because from your because all the cells in your body are coming from your parents. Okay, you're from single. So some of again, I'm just kind of talking offline because I don't know the details, but I would think that some of it is inherited, right? From your parents, right? From your mom, or it is inherited, and that's why when you're a kid, they give you all these shots, okay? So that whatever is not up to that level, right? We can stimulate that. But I would think the original cells, the B cells. So your question is how do you know that it'll have the B cells of the right type, right?
Yeah.
I think that has to do with I mean it's probably inherited. It's already there. And to bring it out, they're giving all those shots. Right?
So if a virus has not been in the generation before, that's the problem to the body that it cannot
Yeah,
resist into
yeah,
it.
yeah, yeah. If a virus, I mean, and that's the problem. That's why they talk about the superbugs in hospitals and all that.
Yeah.
Even the antibiotics will not work against them because those superbugs have figured out how to beat the antibiotics, right? That's why you hear about all this avian flu and things like that, right? So that could be a pandemic, okay? It could kill like millions of people. It can happen because if you don't come up with the antibody and the vaccine, right, in time, you're basically dead, right? So all it will take is that one virus or on one bacteria to, just go out of control. Yeah. And living organisms basically adapt. Okay, so no matter what you do, right? If you're killing the bacteria, the ones that survive, they are resistant now to the antibiotic that you apply. Okay, you apply another antibiotic, the ones that again survive, they are now resistant to the first one and the second one, right? And you go on and on. And so that's why they say that antibiotics should not be prescribed indiscriminately, right? So yeah. All right. Any other questions?
Yeah, could you explain again how the B cell got triggered?
Oh, B cell got triggered. See, what is going to happen is that the B cell, all right, on its membrane, all right, it has that Y sticking out. Okay, it's like a transmembrane thing, all right. So now, when the particular antigen, okay, the one which matches that antibody, it comes and binds the antibody on the cell surface, that will trigger a reaction inside. It'll trigger a reaction. It's a signal transduction cascade that we will talk about. It'll be something is adding some phosphate group here, but at the end of the day, what is happening is there's a lot of signaling that goes on. At the end of the day, the cell gets the signal to multiply, right? And so many copies of that B cell will be produced, and more of these antibodies will be there. Now, antibodies can also be used to purify molecules because they will bind to the antigen with great specificity, right? So, for example, in immunoaffinity column chromatography, right, a mixture of, let's say, the antigen A and other molecules is passed through a column that contains beads that are coated with the anti-A antibodies, right? The antibodies that match A. So what is going to happen? A is going to bind very tightly to those beads. Okay, everything else will just flow through, right? So the flow through the column is going to be discarded, and the A molecules can be recovered by appropriately treating the beads, because the beads now have the antibody that is tightly bound to the A molecules, right? So so that's immunoaffinity column chromatography. I will talk about column chromatography in a few minutes, right? And there can be different kinds, but this is based on immunoaffinity, where you're using an antibody. Another example is in immunoprecipitation. You could take a mixture of molecules that contain A, right? And you're trying to isolate the A. So you can treat that mixture with anti-A antibodies, right? So then A and the antibodies for A will bind very tightly, right? And you can now collect them by centrifugation, which is another technique that we will discuss in a few minutes. Or even by other techniques, you can separate it out from the rest of the mixture. Now, monoclonal antibodies are basically antibodies of just one type, okay? So these are large quantities of a single type of antibody molecule, right? If you want to produce that, again, the producer for that antibody is the B cell that carries that antibody. If you stimulate that to divide, right, then it is going to produce more of that antibody. So what you could do is you could take a B cell, right, from an animal that has been injected with the particular kind of antibody that you're interested in, I mean with the appropriate antigen, right? And you take that B cell and you fuse that with the B cell tumor, right? Now the cell that is taken from the tumor has got the property that it is going to divide uncontrollably. It's going to keep on dividing, right? Whereas the B cell that you have taken from the animal injected with that antigen has the property that that cell is going to produce the appropriate antibody, right? So if you fuse the two together, you now have a B cell that is going to divide uncontrollably, right? And at the same time produce that antibody, right? So you can produce large quantities of that antibody. And again, what I'm saying, to accomplish that in the lab, that's probably a pretty elaborate procedure. Temperature, pressure, I mean, lots of things are there. Okay? And as an engineer, I should not even try to do that. I'm trying to give you the big picture, right, of what's done. Now, antibodies can be used as molecular tags, right, or molecular markers. One could, for example, couple a set of antibody molecules to a fluorescent dye, right? Then you can follow the antibody because, wherever you see that fluorescence, right, that's where the antibody is, right? Or you could attach it to a colloidal gold particle or some other special tag. So for example, in this picture at the bottom, you have some specific antibodies against antigen A. You couple them to fluorescent dye, colloidal gold particle, or some other special tag. Now you have labeled antibodies, right? So you can see. I mean, if it's a mobile mixture, you can see where the antibodies are going. Like inside the cell, you have so many different compartments, right? I mean, if you want to track a reaction, okay, where is that reaction taking place, right? So if you know that an antibody is taking part in that reaction, label it with something, then you can trace its movement, right? All kinds of things you can do like that. Now, before you can study proteins, all right, you would have to isolate them from cells because the proteins are located inside the cell, right? There are transmembrane, all kinds of proteins. Okay, so how do you isolate proteins from cells? So the first step is a cell is enclosed inside a plasma membrane, right? So you have to get rid of that membrane, right, without messing up everything, right? So the first step is to break open the cell by disrupting its plasma membrane, and different techniques can be used. 4 of the commonly used ones are as follows. Number 1, break cells with high frequency sound, right? And don't ask me what the frequency is. I don't know. Okay, I don't do those experimental procedures, but that's one of the techniques that can be used. Another one is use a mild detergent to make holes in the plasma membrane, all right? So the plasma membrane again is a lipid, right? So this could be similar to what you do when you're trying to clean up your dishes, that are soiled with oil, right? I mean, you try to get rid of the oil. So something like this. Then you could force cells through a small hole using high pressure, right? And again, all this has to be done very carefully under the right conditions, right? Because you just want to get rid of the membrane and collect all the contents. The fourth method would be shear cells between a close-fitting rotating plunger and the thick walls of a glass vessel. Okay. How close-fitting, right? How quickly rotating? How thick should the walls be? Okay, those are all pertinent questions, right? I don't have the answers to those, but the people who use these procedures, they would be able to tell you what the experimental protocol is. But that's how it can be done. Now the net result is that the membrane is gone, right? And you have this thing that is called the homogenate or the abstract. That is going to contain molecules from the cytosol, that is the interior of the cell, as well as all the membrane-bounded organelles, right, like the nucleus, the chloroplast, the peroxisome, and so on. Now, if you're careful, right, when you're doing this homogenization, when you're breaking up this membrane, it can leave the membrane-bounded organelles intact, right? And you would like to do that because you want to study something that is going on in that organelle, right? So the next step is to try and separate out all the different components, okay? How do you do that? There are a number of procedures that are used for separation, such as centrifugation, and I will talk about them in the next few minutes, velocity sedimentation, and equilibrium sedimentation. So first, let's look at centrifugation. So in centrifugation, what you're going to do is you're going to take that mixture, put it in a test tube, right, and rotate it at very high speeds, right? So the acceleration that you get is of the order of 10,000 times the acceleration due to gravity, 10,000 g. So obviously you cannot do it in a regular environment, right? You have to have it, very well protected, right? With a lot of metal and other things, and even if the heat is generated, that has to be contained. So there are special centrifuges that are available for this, right? So the homogenate is placed in test tubes and rotated at really high speeds in a centrifuge, right? And the components will separate out mainly on the basis of their size and density, right, in the test tube. And what you can do is, once one component is separated out, you can collect it. You can take it out from the test tube. Then again, whatever is left behind, right, which is called the supernatant, you again centrifuge it at a different speed, right? Again, achieve some more separation. So you can do progressive centrifugation at higher and higher speeds, and you can separate the cell homogenate into its different components, right? And this is the picture. You have a test tube. You have centrifuged the contents of this test tube at a high speed, and you have some pellets that have accumulated at the bottom, and at the top you have the supernatant, right? So you can go ahead and drain the supernatant, and then in another test tube, and then centrifuge it a second time at a different speed to achieve further separation. So that's the first method, that's centrifugation, right? The second method is what is called velocity sedimentation, right? Now subcellular components, that means the different components inside the cell, like these different organelles and all that, they are carefully layered over a dilute salt solution. Now these components sediment or settle down to the bottom at different speeds according to their size, right? So when sedimented through such a dilute solution, different cell components will separate out into distinct bands, right? And then you will collect them after an appropriate time. So you'll have some idea, you have some knowledge about how long you need to wait, then you'll collect them and the components are separated. Another sedimentation procedure is called equilibrium sedimentation. And this procedure can be used to separate cellular components on the basis of what is called their Bowen density, right? Independently of their shape or size, and it makes use of a centrifuge, right? And here the sample is usually layered on top of a steep density gradient that contains a very high concentration of either sucrose, right, which is one of the sugars that we have encountered, right? So sucrose is made up of what? Glucose and fructose, good. Yeah, so you're studying, very good. So high concentration of sucrose or cesium chloride. Again, I mean those chemicals have the necessary property, right? Whatever is needed for this thing to be successful. So each subcellular component will move up or down when centrifuged until it reaches a position where its density matches the density of the surroundings. Remember, there is a density gradient that has been established. Right? So where its density matches, then it's not going to move. Right? So again, a series of distinct bands will form inside the test tubes, and these bands can be separated out by collecting from the base of the test tube. Okay, now you might wonder, why are we going through all these things, right? The reason is that if you read any paper, right, that even uses informatics approaches in the context of medical or agricultural applications, right, they will use some of these procedures over there, right? There will be a section in those papers that is called materials and methods, typically where they will discuss the details, right? What concentration they used, okay, which are the procedures, did they use centrifugation and all that. And I don't want you to be in a position after having taken this course that, seeing that section, it throws you off. You will not understand all the details of the procedure, like, I mean, what solution they use and all that stuff, but you'll have the big picture, right, of what they ran in order to get the data that they worked on and you might want to work on. Right? And I think that is important. That having that domain knowledge is important because otherwise you'll get thrown off when people start using their jargon about column chromatography, gel electrophoresis, and so on. Right? It's important. But again, I mean, this is a big picture. It's a bird's eye view. Right? We cannot go into the details of that. Otherwise people would not be spending semesters and years trying to get a degree over there. Right? So, but we need the big picture. So how do you separate out a mixture of proteins? Now proteins are very diverse molecules. They differ from each other by size, shape, charge, hydrophobicity, and so on. Right? So you can use any of these properties to separate out proteins, right? So that they can be studied individually. And two of the techniques that are widely used to study proteins are column chromatography. We've already seen that. We already saw that immunoaffinity, column chromatography, all right? We saw that, all right? But column chromatography is really more general, right? It's more than just putting some beads with antibodies and trying to separate things out using antibodies, all right? So column chromatography is one of them, and the other one is gel electrophoresis. These are experimental procedures that can be used to separate out a mixture of proteins, right? And let's see how they work, right? So in column chromatography, right, what you're going to have, and I'll go back to the textual stuff a little bit later. So you have a column like shown on the, in the picture on the left, all right, and you have some solid matrix, right? That matrix could be different things. It could be like the beads coated with antibodies, or it could be something else, right? And I'll talk about the different possibilities in a few minutes. So you will apply the sample at the top, right, and you will force it through this matrix, right? Now obviously you put in a special matrix that will pick up, some component of the sample that you're applying, right? And then later on, the rest of the stuff will flow through. Something will get stuck to that column. The rest of the stuff will flow through. Whatever is stuck to that column, you have to have some technique for separating it out from the column later, right? That's the big picture of column chromatography, right? In one case, it could be like antibodies, beads coated with antibodies, where you're trying to, isolate a particular antigen, right? And then you have to figure out how to separate that. So let's just go back a couple of slides. So as I showed you in the picture, the mixture of proteins is forced through a vertical column containing a matrix, and the sample is positioned on top of the matrix, and solvent from a reservoir is used to force the sample down the matrix, right? And different choices for the matrix will lead to different kinds of chromatography, right? In ion exchange chromatography, you're going to separate on the basis of charge, right? So the matrix will consist of either positively or negatively charged beads, which will retard proteins of the opposite charge, right? And thus separation in this case is achieved on the basis of charge, right? Then in gel filtration chromatography, the matrix will consist of tiny porous beads, right? Really small porous beads. So protein molecules that are small enough to enter the holes in the beads, they'll be delayed, okay? And they'll travel more slowly through the column. The larger protein molecules will just go through. So in this case, the separation is achieved on the basis of size. And in the last case, which we have already seen, in affinity chromatography, the matrix consists of molecules that interact specifically with the protein of interest, for instance, an antibody, right? So the molecule will get stuck to the antibody and the rest of the solution will go through, right? And finally, once the flow has stopped, right, things have been forced through the column, then the proteins that bind specifically to such a column can be released by changing the pH, like the acidity or alkalinity, or by concentrated salt solutions, and they emerge highly purified. Right? So you've separated out the mixture of proteins. So this is one method, column chromatography. Any questions? Another method that is used to separate out proteins is called gel electrophoresis, and this should be, uh, the will find that very natural, because if you're separating proteins on the basis of charge, you apply an electric field, all right? Negatively charged molecules will move towards the positively charged end, and you'll separate them out, right? So this procedure is used to separate it can be used to separate proteins both on the basis of their size and charge, right? And there are two methods here, right? One of them is called SDS page, and again SDS is the abbreviation for sodium dodecyl sulfate and then page is polyacrylamide gel electrophoresis, right? So SDS page, protein separation is achieved on the basis of size, right? And I will explain the details in a moment. And in isoelectric focusing, you are going to separate proteins on the basis of their charge. So let's look at them one by one. So first we'll look at SDS page, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. Again, I mean, that's the particular compound that is needed for running this gel electrophoresis. Right? You don't have to memorize that. You know, if it comes on the exam, put SGS page. If you don't remember, that's okay, right? But hopefully you'll pick it up, by reading the notes again and again, right? So the individual polypeptide chains form a complex with negatively charged molecules of SGS, right? So the protein is forming a complex, so the resulting complex is negatively charged. So if you apply an electric field, it is going to move, right? From the negative side of the field to the positive side. So when subjected to an electric field, they migrate as a negatively charged SGS protein complex through a slab of porous polyacrylamide gel, and the proteins will migrate at a rate that is proportional to their molecular weight or their size, right? If it's a bigger thing, it will probably need longer, right? So thus protein separation is on the basis of their size. So that's SJS page. Another method where you can separate proteins on the basis of their electric charge is called isoelectric focusing. Now for any protein, right, you're separating now on the basis of charge. There is a characteristic pH. pH again is the hydrogen ion concentration, right? So there is a characteristic pH which is called the isoelectric point at which the protein will have no net charge, right? And therefore will not move in an electric field, right? So in isoelectric focusing, what you do is that proteins are electrophoresed the same way that you do SGS page, but in a narrow tube of polyacrylamide gel, and here you don't have SDS, you don't have sodium diodecyl sulfate, right? You'll just have polyacrylamide gel, but in that you have established a pH gradient by a mixture of special buffers. So as you move through that gel, all right, the pH is changing, right? So when you apply an electric field, the protein will go to its isoelectric point, and it will not move anymore from there, right? So each protein moves to a point in the gradient that corresponds to its isoelectric point and stays there, thus allowing protein separation on the basis of charge. And actually you can combine these two methods, right, and apply them in perpendicular directions to do what is called two-dimensional polyacrylamide gel electrophoresis, right? So here, first, isoelectric focusing is used to separate proteins on the basis of electric charge, right? Once you have done that, then you can apply SDS page in a perpendicular direction to achieve further separation on the basis of size, right? And the picture would look something like this, right? So in the horizontal direction, you're doing isoelectric focusing where you've established a stable pH gradient, right? And you will separate on the basis of charge. In the vertical direction, you're doing SDS page, right, where you're separating on the basis of size, right? And this information on this gel, all right, they will, there are things called, different kind of blots like southern blots, northern blots, and so on. What they will do is they can actually apply blotting paper on it, right, and capture this information, right? Then, in addition, if some of the proteins you label them using an antibody, right, or fluorescent dye, you could take pictures and figure out, where the protein has gone. So there are different combinations of these methods used with all this blotting and all that, and they have different names. Even for DNA, you can do the same thing. We will talk about that later, right? This kind of electrophoresis to separate out DNA on the basis of size. You can do that and capture that onto like blotting paper, right? And these days, actually, they have methods for, actually not using blotting paper and messing around with that, capture that information onto something else and then process that, send it to your computer and process it and so on. Okay. All right, any questions? So far we have discussed protein separation, right? How do you take the cell, break it up, take all the proteins out, and then separate them out on different bases, size, charge, and so on. Okay, and use all these methods. Now, how do you determine the structure of a protein? Again, I mean, we said that, the structure of a protein can be determined at different levels. What is the first level? Primary structure, which is the amino acid sequence, right? Then the second level is alpha helix and beta sheet, right? The third level is three-dimensional conformation, and then the fourth level is where you have proteins that are made up of multiple units, okay, the quaternary level. So here we are focusing on the first level. See, the second and the third level is not that easy, okay? We do not know, right, how to go from the amino acid sequence to, let's say, the alpha helix, beta sheet, and then maybe parts of it are known, okay, in some specific cases they know, but there isn't a good method to go to the three-dimensional conformation because if we could do that, that would be really a big breakthrough, right? So if anybody wants to work on that, here's the problem, right? Not necessarily for a PhD, but it is a grand challenge. If you can predict that, then we would have made a big advance in molecular biology, drug design, and so on. Okay. So how do you determine the primary structure? So in the first approach, a reagent is used to isolate the N-terminus. Again, remember, a protein is made up of amino acids that are linked together. When you link these amino acids together by peptide bonds, right? At one end you will have the N-terminus, the amino terminus or the N-terminus. The other end you'll have the carboxyl terminus, right? Or the C-terminus. So in the first approach, a reagent can be used to isolate the N-terminus amino acid from the rest of the polypeptide chain. Like you might know that there's some reagent that will go and chop off the N-terminus amino acid, and then you try to figure out what that amino acid is. So the isolated amino acid can then be identified using a full set of known amino acid derivative standards. Example, the position to which a particular amino acid migrates under a SDS page. Like you could isolate this, then see where it migrates, right? Then you have the list where the 20 migrate, and then figure out which amino acid it is. But this is going to be a laborious procedure, right? And you can use it repeatedly to determine the entire amino acid sequence of the protein. Right? But if it's 250 units long, you'll have to do it 250 times. However, it is often found that the amino terminus amino acid is chemically blocked. Right? It does not respond to that chemical. And in that case, you cannot use that method. And there's a reason for it. The cell is producing the protein, right? For a purpose. It's not producing that protein so that you and I can go in there and cut out the N-terminus and then figure out what that sequence of amino acids is. It's for a function. So the cell is going to try to protect that, right? So it might be chemically blocked. You cannot go and take it out. Right? So in that case, you cannot use this particular method. Now the second approach for determining the primary structure of a protein makes use of the fact that there are certain chemicals which will selectively cut proteins, cleave proteins, at particular amino acid locations. For example, there is an enzyme that is called trypsin, which is going to cut on the carboxyl side of lysine or arginine residues. Right? Again, we talked about 20 different amino acids. Lysine and arginine are two of them. Right? So if there is a protein, you have a lysine, it will cut on the carboxyl side. Remember, there is a carboxyl side, there is an amino side. Okay, on the carboxyl side, it will cut it. Right? While there is another chemical that is called cyanogen bromide, which will cut peptide bonds on the carboxyl side of methionine. Again, methionine is another one of these amino acids. Right? So by treatment with these reagents, you can get smaller proteins or smaller peptides that you might be in a better position to analyze. Right? So these reagents can produce a few relatively large peptides, and these can be separated using, for instance, again, gel electrophoresis to create a peptide map which is diagnostic of the protein from which the peptides are generated. And the sequence of overlapping peptides can be used to laboriously piece together the sequence of the original protein. Again, it's not easy because you've cut it, you just got those pieces. It's like putting a puzzle together, right? But if you do enough number of times, you probably can. Okay, you have enough redundancy and you can put it back together. So now, again, this is not very high tech. Now we go to the high tech method, right? And that makes use of recent advances in DNA technology. Remember, I said earlier the DNA codes for proteins, right? The information on the DNA AG, CT, that actually codes for proteins. That is first transcribed into RNA and then translated into protein. So for each amino acid, we know what the code is. Okay, what sequence of letters codes for a particular amino acid, or what sequences of letters code for a particular amino acid, we know that. Okay, so what you could do is, suppose there's a protein, you do not know the entire amino acid sequence, but you know, let's say, about 20 of the amino acids, right? So from those 20 amino acids, you could figure out, what should be the letters in the corresponding DNA. Right? At least in the RNA you can figure out, and then try the DNA. So with that, you can take the complement of that. Okay? So you got these 20 amino acids, you went ahead and figured out what the corresponding DNA code would be for just for those 20 amino acids. Right? And then you create the complement of that code. Okay? Create a DNA strand that is the complement of that. Then if you mix it up with the appropriate tissue and all that, you'll be able to pick up, right, the entire chain, the DNA that codes for the entire protein, right? Just by matching up the DNA corresponding to those 20 amino acids. Once you pick that up, then you can sequence that DNA and figure out, what the entire sequence is, right? right? So once you sequence that, now you can take them like 3 at a time and figure out what the different amino acids are. Okay, so this is a technique that is made possible by current day DNA technology, which we will discuss in detail a little bit later in this course. Right? So there is a code that is called the genetic code, which we will talk about in a few lectures, right? That basically maps the sequence of DNA and RNA to the corresponding amino acid, right? And this code is universal. This genetic code is universal. All living organisms on this planet, they use this genetic code, right, for making proteins out of from DNA. So in addition to the above approaches, the three-dimensional structure of protein molecules has been studied using X-ray crystallography and nuclear magnetic resonance spectroscopy. So you can see that chemists and physicists and all are also involved. Okay, so they are going to go directly to the three-dimensional conformation, right, which is really important for us. But if somebody could figure out how to go from the primary sequence to the three-dimensional conformation, right, which I suppose is the conformation of lowest energy, right, that would be a big, big scientific breakthrough. Okay, any questions so far? Now we'll talk about proteins that are enzymes, right? See, we talked about enzymes in the last chapter, right? That enzymes can speed up reactions by as much as 10 to the power of 14 and all that, you know. So now we want to discuss a little bit the properties of enzymes and introduce some terminology that is associated with enzymes, right? Or that you encounter in the study of enzymes. Now the binding strength between an enzyme and its substrate, or an antibody and its antigen, right? Again, these are like two proteins that are binding to each other, is measured by what is called the equilibrium constant K, right? So again, look at the reaction. Suppose you have two things, A and B, that react together to produce the product AB. Okay, so this is an association reaction that is proceeding from left to right. So for this reaction, the reaction rate, which is called the association rate, is given by association rate. See, we know that the rate of reaction is proportional to the concentration of the reactants, right? You know that, right? So the association rate is going to be equal to the association rate constant, which is the constant of proportionality, right? K on, multiplied by the concentration of A times the concentration of B. Okay? Where A within square brackets denotes the concentration of A in moles per liter, B within square bracket indicates the concentration of B, right? So that is the rate at which the forward reaction will take place, right? But then you also have a reverse reaction where the products are going to dissociate to produce A and B, right? And what I'm covering here, it applies to other reactions too. We've just picked an association reaction just for the sake of simplicity, right? So the reverse reaction is that the product AB, original product AB, is dissociating into the two components A and B. And for this reaction, the dissociation rate, right, which is the rate of the reverse reaction, is given by dissociation rate equals the dissociation rate constant, again the constant of proportionality, which we will call K sub off, times the concentration of the product. Okay, because the concentration of AB, because that is what is driving this reverse reaction. Now, when the equilibrium is reached, right, then the two rates have to be equal, right? As much of reactants are get converted into products as products are getting converted into reactants, right? So you would have to have K on times A, the equilibrium concentration of A multiplied by the equilibrium concentration of B equals K off multiplied by the equilibrium concentration of the product AB. Agree? So then the quantity capital K, which is basically K on over K off, which is the concentration of the products divided by the product of the concentration of the reactants, right? That is called the equilibrium constant. And you know that from high school chemistry, right? You've seen that before. Here I'm going to try to relate that to free energy also, right? So for a particular chemical reaction, if K is large, right, if the equilibrium constant is large, then this equation tells you that the equilibrium concentration of the product AB will be much larger than the equilibrium concentration of the reactants A and B, right? Which means that the reaction will have a strong tendency to proceed in the forward direction, right? A plus B to AB, right? And this relates to free energy. That means that means the reaction is happening with a decrease in free energy. If you remember, last time I had written down a formula for that free energy change, the standard free energy change associated with the reaction, right? And there was that term, that concentration dependent term with a minus sign in front, right? with the product at the top and the reactants at the bottom. Natural log over there. So, if the concentration of the equilibrium concentration of the product is much higher than that of the reactants, right? Then you will get a large negative free energy change, right? And the reaction is going to be energetically favorable. So I hope this ties in with what you learned in high school, right? About the direction in which the reaction is going to go. Any questions? Yeah.
Question. So, larger K means basically the concentration of AB is greater than A and B individually, right?
Right.
And the reaction usually proceeds in the direction in order to reduce the concentration.
No, this is equilibrium concentration. Yeah. What I'm talking about is that the equilibrium concentration of AB will be much larger than the equilibrium concentration of A and the equilibrium concentration of B. Right. Yeah, so the reaction will proceed at equilibrium. AB will have much higher concentration than either A or B, which means that the reaction is proceeding from left to right.
Okay, yeah. So eventually it should also go backwards, right?
No, it is going backwards all the time. Okay.
Okay.
You are having reaction going left to right, right to left. At equilibrium, the reaction, at equilibrium, nothing is changing. Okay. So the concentration of AB and A and B stay the same because, if A and B goes to the right, the same amount of AB moves to the left. Right. That's what defines equilibrium. Yeah. But it's happening. It's a dynamic equilibrium. It's going in both directions. Yeah. And if this value of K is large, that means at equilibrium the concentration of AB will be much higher than the concentration of the reactants, right? And the reaction is energetically favorable. It's accompanied by a large negative free energy change, right? If you look back at that formula, I don't have it here right now, or pulling it, I'll have to go through 50 slides, to pull that up, but it's there in your notes, right? So it is energetically favorable, right, in the direction from left to right. Now enzymes, as I said last time, they have powerful and highly specific catalysts, right? And each enzyme will catalyze only a very specific chemical reaction. And enzymes that catalyze similar reactions, they can be grouped together and they have special names, right? And these names could throw you off. If you look at a textbook on biology or you try to read a paper, right? That is at the interface of engineering and biology. Hydrolases, these are enzymes that catalyze a hydrolytic cleavage reaction, right? What would be a hydrolytic cleavage reaction? What is hydrolysis? Breaking down with water, right? And what is it? Is the reverse of what? Condensation reaction, yeah. So any enzyme that catalyzes hydrolysis, right, that would be called a hydrolase. Okay. Nuclease, okay. Nucleases are enzymes which break down nucleic acids because remember, to form nucleic acids, you have to link nucleotides together by phosphodiester bonds, right? Remember I showed you the nucleotide unit, and again you'll see more of this later in the next chapter on DNA and all that. We will see the details of how everything is linked together, but I kind of gave you a preview, right, or a glimpse of how nucleotides link together. They're linked together by phosphodiester bonds, right? So nucleases basically disintegrate nucleic acid, right? So these are enzymes that will break down nucleic acids by hydrolyzing. Again, this is also a hydrolase, but it's more specific, right? Because it's going to break down nucleic acid, right? So hydrolyzing phosphodiester bonds between nucleotides, right? Then proteases, all right, you must have guessed, all right, these are enzymes that break down proteins, okay. Again, there is hydrolysis, all right, because the condensation reaction linked together the amino acids to form proteins, right. The reverse is hydrolysis. So proteases are enzymes that break down proteins by hydrolyzing peptide bonds between amino acids, right. Synthases, all right, or sometimes they're called also synthetases, all right. These are, so that's the general name for enzymes that synthesize molecules in the anabolic reactions by condensing two smaller molecules together. Like proteins are formed by some synthase, all right. DNA is formed by DNA synthase, all right, right, or DNA, what they call DNA polymerase, all right. Isomerases are enzymes that catalyze the rearrangement of bonds within the single molecule. So they will form isomers. So from one isomer, let's say glucose will go to mannose, all right, or glucose will go to galactose, all right. That is going to be brought about by an enzyme that has the appropriate name, all right. let's say glucose galactose isomerase. Okay, I haven't seen that enzyme, but I'm saying those are the kind of names that you will see, right? Polymerases are enzymes that form polymers, so that catalyze polymerization reactions such as the synthesis of DNA and RNA, right? So if you have these nucleotides, you want to form DNA from nucleotides, that is done by DNA polymerase, right? You want to form RNA from RNA nucleotides, all right? You will the enzyme that is needed is RNA polymerase, and we will see all these enzymes within the next two chapters, right? We will come across them. Okay, any questions so far? So you can think that polymerases are also synthases, right? I mean, they synthesize something, right? But this is more specific. It forms that polymer like DNA or RNA and so on. Okay, next one is very, very important, right? Kinases are enzymes that catalyze the addition of phosphate groups to molecules, right? See, so kinases, they'll add phosphate groups. Phosphatases will remove phosphate groups, right? These are very important because, you know, the way our cells are controlled is by kinases and phosphatases. Okay, if your cell has to divide, remember I said, I think one of you asked me, was it you, that how the B cell is going to divide, right? You asked me, right? I mean, so something is going to come and bind that antibody from outside. It is going to signal inside. I talked about signal transduction. What is that signaling going to be? Most of it is going to be kinase activity. Something will add a phosphate group to some other molecule. Now, if you add a phosphate group to a protein molecule, the phosphate group has got two negative charges. Because of that, the shape of the protein will change. Okay, so and it can go from active to inactive conformation. In fact, all our cells, they control their activity by this kind of behavior, or kinases and phosphatases. Phosphatases, they determine whether your cell is going to divide or not. And let's say if somebody got cancer, it could be that some kinase inside, right, is acting up, right? It is just phosphorylating something downstream. It's putting a phosphate group, even without receiving the stimulus from upstream, from the cell membrane, right? And that could cause the cell to divide. So kinases and phosphatases are extremely, extremely important, right? I mean, it's not obvious, at least for the kinase. Okay, how do you figure out that that adds a phosphate group? You know, it's not sounding like polymerase. Like phosphatase, I can say, yeah, it takes off phosphorus. But kinase is very important, and a lot of the anti-cancer drugs, these are kinase inhibitors, right? They will go and stop the kinase from adding the phosphate group. So this is important. Oxidoreductases is a general name for enzymes that catalyze reactions in which one molecule is oxidized while the other is reduced. Right? I talked about oxidation and reduction earlier, and I said they occur together, so you have what are called redox reactions. Right? So oxidases are things that bring about oxidation, reductases are things that bring about reduction. Right? And enzymes of this type are often also called oxidases, right? Then reductases, and sometimes they are also called dehydrogenases because they remove hydrogen, right? Like if somebody drinks alcohol, that has to be converted into acetaldehyde by an enzyme that is called alcohol dehydrogenase, right? So these enzymes are important. Right? And the nomenclature in biology is very systematic. You know, if it oxidizes something, it will be called that thing oxidase and so on, right? Something reductase. So at least that, you know, the biologists have done for us. You know, otherwise there's no way we could follow the biology literature, right? Uh, Another class of enzymes which are very important are what are called ATPases. These are enzymes that hydrolyze ATP. Remember, I talked about ATP, adenosine triphosphate, three phosphates, right? So you hydrolyzing ATP means breaking the last phosphate bond to get ADP, right? Now, ATPases typically are enzymes that produce some kind of energy, right? So many proteins have an energy harnessing ATPase activity because you have to burn up energy, all right, to let's say make the muscle move, right? So many proteins have an energy harnessing ATPase activity as part of their function. For example, motor proteins such as myosin, which is the muscle protein that is responsible for generating movement. Then you have membrane transport proteins, like the cell has to transport sodium out of the cell, take potassium inside. Whenever you need to transport something against the concentration gradient, right? Let's say the concentration inside the cell of sodium is not that high, but still you want to keep pumping sodium out, right? If it's against the concentration gradient, you have to do work, right? I mean, if you're trying to move uphill, you have to do work, right? You need energy, right? So those pumps, the sodium potassium pump, right? They have an ATPase activity. Like they will, use up ATPase as part of their enzymatic activity, right? So they'll break up ATP. So they are called ATPases. All right, so now, okay, any questions so far? So now I want to talk about reaction rate versus substrate concentration, right? So suppose you have an enzyme and it is processing the substrate to give you the products. So as the substrate concentration is increased, the reaction rate initially goes up as more substrate molecules are available to interact with the enzyme, right? But beyond a certain substrate concentration, the reaction rate saturates because the enzyme is not able to handle any more substrate, at the same time. So the enzyme is already functioning at its maximal speed and cannot process substrate molecules any faster. This limiting velocity of the enzyme is called Vmax. Okay. So again, this is important. Keep that in mind. Okay. When biologists talk about the activity of enzymes, they will talk about two parameters. One is Vmax, and the other one is Km. I'm getting to Km on the next slide. Right. So so it's not like the activity is described by some equation. Like when we describe an RC circuit, right? The response of an RC circuit, we talk about a time constant, right? I mean, the product RC and so on. Right? They don't describe it that way. They describe it in terms of Vmax and another parameter that is called Km. So and I'll introduce the Km in a few minutes, but look at this picture, right? So along the x-axis you have the substrate concentration, right? And along the y-axis you have the rate of reaction, right? Or the velocity of the reaction. So as the substrate concentration increases, the velocity of the reaction is increasing, and then it saturates at Vmax. Okay? Now the substrate concentration at which the reaction proceeds at half the maximal velocity, right? That means 0.5 Vmax, right? That substrate concentration is called Km, right? Or it is called the Michaelis-Menten constant, right? So these are the two parameters that they will specify for a substrate, right? For any enzyme, right? So now these things actually determine, right? So suppose you have a number of substrates that are competing for the same enzyme, right? Number of substrates. So which reaction will go forward, right? That depends on the values of the Vmax and Km of each of the substrates, right? Now if the substrate, if there's an unlimited amount of substrate, right? If you have lots of substrate, right? More than what the enzyme can handle, which number do you think is going to determine the rate of the reaction? Is it going to be Vmax or is it going to be Km? Make a guess, No, you have too much of substrate, right? It's going to be the Vmax. If the substrate is not limiting, right, it'll be Vmax. So the one which has the higher Vmax, that's the reaction that will, be preferred, right? Right. On the other hand, if the substrate is a limiting factor, right, this Km actually denotes the affinity of the substrate for the enzyme. The one with the smaller Km, that is the one that will get the enzyme, right? So that's again a general principle, right? So the substrate concentration at which a given enzyme can function at half its maximal velocity, that is called Km for that enzyme, and it measures the affinity of the particular enzyme for the substrate. And if two enzymes compete for the same substrate, then the values of their Vmaxs and Kms determine which reaction gets preference. So if the substrate is limited, then the enzyme with the lower Km gets preference, right? Because you're operating in a different region of that curve, right? On the other hand, if the substrate is not a limiting factor, you have tons of substrate, then the enzyme with the higher Vmax is going to dominate. Now, despite their versatility, there are instances where a protein needs the help of a non-protein molecule for performing functions that would have otherwise been impossible. And I give you a few examples of that. A molecule of hemoglobin needs the help of 4 heme groups. These are ring-shaped molecules containing iron in order to be able to perform its function of picking up oxygen in the lungs and releases it in the tissues. In fact, the heme group is what basically gives blood its red color, right? The iron in there, right? Another example is the signal receptor protein rhodopsin in the retina of the eye detects light by means of a small non-protein molecule that is called retinal, which is embedded in that protein. Right? So all the proteins are very versatile. They perform most cellular functions, but there are instances where protein needs help from a non-protein molecule to perform its specific function. All right, any questions so far? So now let's talk about the regulation of catalytic activities of enzymes. The cell can regulate how much of each enzyme to produce by regulating gene expression. And we will talk about that in the next chapter or the chapter after that. Okay. Gene expression means basically the corresponding part of the DNA is copied into RNA and then to be translated into protein. The cell controls that very tightly. The cell can also control enzymatic activities by confining sets of enzymes to particular subcellular compartments. The cell can do compartmentalization, right? Because if you confine an enzyme to a particular compartment, then that enzyme can be active only in that compartment, right? You don't want it elsewhere. An enzyme also can change its activity in response to other molecules that it encounters in the cell, right? For example, if the level of glucose, right, in the environment is low and you only have lactose, then you need the enzyme that can metabolize lactose, right? Having the enzyme that can metabolize glucose is not going to do you any good, right? And the cells do respond adaptively to these kind of situations. An example of an enzyme changing its activity in response to the presence of other molecules is feedback inhibition, right? And this, I mean, for those of you that have taken control systems, this is like a feedback control diagram, right? So here you have a molecule X that produces Y, right? Which in turn produces Z, right? And the enzymes are marked as E1, E2, E3. So you have W, right? Then E1 produces X, that produces, then E2 produces Y, then E3 produces Z from Y. But then the product of that last step, right, is actually inhibiting that original enzyme E1, right? So that is like a negative feedback loop, right? So in feedback inhibition, X produces Y, which produces Z. However, Z inhibits the production of X. This is an example of what is called negative feedback, right? It's single loop, and people in the controls area, they've seen this all the time. The challenge here is that in biological systems or in living organisms, there are multiple loops like that are interacting with each other, all right? So it's a multi-input, multi-output, nonlinear system, right? So that's complex. Another example of negative feedback in a cell, when there is a rise in the ADP in the cell, again, adenosine diphosphate denotes a low energy state, right? So does adenosine monophosphate, right? And I talked about one molecule last time, right? What is the molecule that indicates the low energy? Cyclic AMP, that's another indicator. So anyway, when there is a rise in ADP in the cell, that indicates a low energy status, all right? Then what do you need to do? You need to bump up the energy, right? So you need to burn more glucose, right? Oxidize glucose. So several enzymes that are involved in the oxidation of sugar molecules, they are activated. So that is going to, and once the sugars get oxidized, a lot of the ADP is going to get converted to ATP, right? So this will stimulate the cell to convert ADP to ATP. This will lower the level of ADP in the cell and it will restore the desired energy level, right? So this is also an example of negative feedback. Although the biology book mentions this as a positive feedback, but I think they got their terminology wrong, right? They're not controls engineers, right? So. Next, I want to talk about, I'm almost done with this chapter, just a couple of things and we'll get done. I probably might have to go 5 minutes over, but we'll get done today. Now, see, in feedback inhibition, oftentimes it is found that the regulatory molecule has got a shape which is totally different from the shape of the substrate of the enzyme. Right? So normally you think that the substrate and the regulatory molecule, they're competing for the same spot on the enzyme, and therefore, if you have the regulatory molecule, the substrate it cannot be processed. Right? So, however, in this case, the feedback inhibition is not caused by the regulatory molecule binding preferentially to the same binding site on the enzyme. Instead, the enzyme here has got two binding sites, right? One for the substrate, one for the regulatory molecule. And these proteins are called allosteric proteins. So when the regulatory molecule comes and binds the protein at one site, right, it slightly changes the shape of the protein, because of which the substrate cannot come and bind at the other side because the shape has been altered, right? And this was quite puzzling to scientists initially, but finally they figured out, this kind of allosteric protein activity. Now, as I said before, eukaryotic cells, that means, let's say cells in humans, cells which have a nucleus, they commonly regulate a protein's function by adding a phosphate group covalently to one of its amino acid side chains. Right? This is something I've already mentioned. Because each phosphate group carries two negative charges, the enzyme catalyzed addition of a phosphate group to a protein can cause a major conformational change. And removal of the phosphate group by a second enzyme returns the protein to its original conformation and restores its initial activity. So you can turn a protein on or off by just adding a phosphate group. So kinases and phosphatases are very, very important, right, in eukaryotic cells. So in eukaryotic cells, protein kinases and protein phosphatases play an important role in switching proteins from inactive to active conformations and vice versa. For example, proteins undergo dramatic conformational changes when they bind GTP. Again, GTP is guanosine triphosphate, just like adenosine triphosphate ATP, except that instead of A you got a G, right? Now the protein bound to GTP is active. It's like it's hyperactive, right? If it's bound to GDP, it's inactive, right? So what is going to happen is that when to activate that protein, it will bind GTP, but that GTP is slowly hydrolyzed, right? And then it becomes GDP, the protein becomes inactive, right? Now in cancer and all, you can have situations where it is bound to GTP, right? And then the GTP is not hydrolyzed, so the protein is permanently active, right? And that can lead to excessive cell proliferation in cancer. So after a while, the bound GDP is released, but the protein remains inactive until it binds GTP again, right? Again, guanosine triphosphate. Now I want to just finish this chapter by talking about how cells can, how proteins can actually walk like you and me, right? And I think before I go through this text, let me just explain it through the picture, right? So here you have a protein that is on the left that is trying to walk, right? So now usually what will happen is it'll walk to the right, to the left, right? It's going to be like a random walk, right? So there's not going to be a net movement in either direction. So how do you make this directed movement? Well, you basically couple the walking, right, with the hydrolysis of ATP. So in the first step, you have the protein in this conformation. In the second step, the conformation has changed slightly, right? It's stepping onto it's making contact. Both the feet are making contact. But then there is an ATP. If you can see that, can you see that A and then there are 3 phosphates, all right? In the third step, the rear leg is pulled forward, right? But at the same time, right, when you're going from the third step, uh, there has been a hydrolysis because there is a phosphate that is coming out, right? And in the fourth step, you're back to the original situation, right? However, you walk to the right. Why? And this is an irreversible thing because you have hydrolyzed an ATP. That is an irreversible reaction because it is accompanied by a large negative free energy change, right? So that reaction is not going to spontaneously get reversed. So this is how things like DNA polymerases and RNA polymerases and all, they move along the DNA, right? By coupling their movement to ATP hydrolysis so that they get directed movement, right? And this is what is explained in the text on this page, but I think it's much easier explained with the picture. So motor proteins can produce directed movement in cells by coupling the hydrolysis of ATP to conformational changes in the protein. In the absence of ATP hydrolysis, the protein would move back and forth in different directions, generating random movement. By coupling the movement to ATP hydrolysis, which is an energetically favorable reaction, large negative free energy change, the movement steps are made irreversible, thereby producing directed movement. And as I said, this is the mechanism by which DNA and RNA polymerases move along a template DNA strand. Right? Let's stop here for today. Right? We are at the DNA chapter, which I will cover next time.